Well, good morning, and thank you for joining us for Winship Grand Rounds this morning. If you're an Emory University or healthcare employee and would like to receive CME credit hour for attending today, login information is 523614, which can also be found in the chat feature at the bottom of your screen for our virtual attendees. If you have any questions with this webinar or the CME login, please send Margaret Johns an email or drop a note via the chat feature. This morning, we are pleased to welcome Dr. David Yu. Dr. Yu received his MD from University of Texas at Southwestern Medical School and PhD from the University of Cambridge prior to completing residency training in radiation oncology at Vanderbilt University Medical Center. During his residency training, Dr. Yu was an American Board of Radiology Holman Research Pathway Fellow. Dr. Yu is also the Jerome Landry MD Chair of Cancer Biology and Associate Professor in the Department of Radiation Oncology at Emory University School of Medicine. He serves as the Director of the Division of Cancer Biology and Vice Chair of Biology Research. He specializes in the treatment of head and neck cancer and breast cancer. As a Georgia Cancer Coalition Distinguished Cancer Clinician and Scientist, Dr. Yu's goal is to translate insights gained from interactions with his patients and his research in the laboratory to innovate therapies to improve the quality of care for patients with cancer. Dr. Yu is also a member of several national committees on ASTRO, ASCO, ACR, and RSNA, to name a few. At Emory, he is a member of the Emory Graduate Programs in Cancer Biology and Genetics and Molecular Biology, and the Emory MD PhD program. All this while being a great colleague and friend. Welcome, Dr. Yu.
Thank you very much for that introduction, Jolenta. So very excited to be here to present our data. I just want to start off with a little more about my background. So this is a picture probably a number of years ago. It's my sister, Jennifer Yu. She's a physician scientist at radiation oncology at the Cleveland Clinic. She works on GBM stem cells, treats CNS cancers, and is a co-investigator on our U54 work. My dad was a biochemist, worked on gangliosides, professor for many, many years at Yale University. When I was in 8th grade, we moved to Richmond, Virginia, where he became chair of the Department of Biochemistry, and later director of the Institute of Molecular Medicine at Genetics. He passed away last year at the age of 84 with active R1 funding, and remarkably was continuously funded for 45 plus years, and has just been an inspiration for my sister and I as physician scientist researchers. I have a clinic at Grady where I treat primarily head and neck and breast cancer patients. This is our group here. I want to acknowledge them for providing inclusive excellence. This is our Jerome Landry, our chief for many, many years. Unfortunately, he also passed away last year, and I'm honored to hold a professorship in his name. So our lab is interested in understanding how cells respond to DNA damage. We know the genome is constantly exposed to a variety of agents that challenge its integrity. This includes both environmental agents such as radiation, ultraviolet light, reactive chemicals, and endogenous agents such as reactive metabolic byproducts or depletion nucleotides. The DNA damage response is a signaling cascade that recognizes these challenges and coordinates diverse DNA repair and cell cycle checkpoint pathways. So it's critical for maintenance of genome integrity and avoidance of various diseases, including cancer. So the DDR is critical for cancer treatment because many cancer treatments induce DNA damage and replication blocks. And this includes all types of radiation therapy, IR, electron beam therapy, proton therapy, many types of chemotherapeutic agents. You have alkylating agents, antimetabolites, topoisomerase inhibitors, even novel antibody drug conjugates such as sacituzumab govitecan that has an irinotecan metabolite SN38 conjugated to anti-TROP2 that's expressed on many cancer cells. And SASI has been shown to improve overall survival and metastatic triple-negative breast cancers and even hormone receptor positive breast cancers. So the DNA damage response is critical for prevention of cancer and for governing its response to treatment. So in human precancerous lesions, there's aberrant DNA replication, which induces activation of DNA damage response. So looking at the kind of neoplastic IHC slides here, compare neoplastic tissue versus normal tissue, there's increased markers for P67, a marker for proliferation, as well as GAMH2X and 50BP1 markers for DNA damage response. So the DNA damage response then mobilizes DNA repair, causes cell cycle rest to allow for additional time for repair, or if there's too much damage, induces cell death. So thus, we think of DNA damage response as a cancer barrier. However, this creates a selection pressure for mutations or epigenetic silencing of DNA damage response genes, which ultimately leads to genomic stability and development of cancer. However, the same genetic changes that drive cancer development can also impair DNA repair pathways which may be exploited for cancer treatment. So our lab is a strong interest, longstanding interest in sirtuins. These are interesting proteins. They are NAD+ deacylases that remove these cytomodification proteins to regulate activity. They also have mono ADP ribosylation activity, as well as long-chain fatty acid deacylation activity, the removal of myristoyl succinyl groups. And we have some preliminary data showing sirtuin demyrisylation activity in the DNA response that I'll share. Sirtuins are also active by resveratrol, which is a red wine extract that's been shown to have helpful benefits, as well as caloric restriction, so linking them to metabolism. They're involved in genome maintenance, aging. You can overexpress sirtuins in yeast, fruit fly worms, and mice, and prolong longevity, so you can live longer. Again, linking metabolism and tumor suppression. Knock it out, you can form a number of cancers. So a fundamental principle of cancer is that cancer is ease of aging. So the older you are, more likely you are to get cancer. So these are the ACS breast and cancer facts and figures in 2020 for breast cancer. And you can see the single strongest prognostic factor is age. This is true across all different races. So this is work by Dave Gaius, this lab, and they showed that sirtuin is a breast and liver cancer tumor suppressor protein. So female mice deficient sirtuin develop breast cancers, male mice develop liver cancers, and moreover, sirtuin expression is decreased in human breast and liver cancer, suggesting that sirtuin is a tumor suppressor protein. On the other hand, this is work by David Sinclair's lab, and BubR1 hypomorphic mice, which have premature aging, if you overexpress sirtuin, can prolong longevity. So we think these sirtuins may provide a really intriguing link between aging and cancer might be related. So this is the first postdoc in my lab, Hui Zhang, and first graduate student in my lab, Pam had. So what they did is they silenced sirtuin by short interferon RNA, siRNA, and showed that you have hypersensitivity to hydroxyurea and gemcitabine. They inhibit ribonate reductase between DNPs, also sensitivity to mitomycin C and cisplatin, which are DNA cross-linking agents. We also show in sirtuin knockout cells generated by CRISPR-Cas9, we have hypersensitivity to radiation treatment. We can rescue this by expression of sirtuin wild type, but not deacylated active H187Y, suggesting that the deacylated activity of sirtuin is required for governing resistance to DNA damage. So we next wanted to ask what substrates might sirtuin be regulating. So we used a proteomic approach in collaboration with Nick Seifert and Proteomics Core, using a pan-acidyl antibody. This is a label-free approach, and 8 sirtuin proficient and deficient cells treated with or without hydroxyurea. We identified over 1,200 cell peptides that were enriched. We were interested in those. So a shift in the left indicates a decrease in acetylation, shift in the right increase in acetylation. So we're particularly interested in those in the brown module. You have a decrease in acetylation in response to damage. You can rescue that with sirtuin depletion, suggesting that these are peptides or substrates that are deacylated by sirtuin in response to DNA damage. So over the years, we've identified a large number of DNA response proteins that are deacylated by sirtuin-2. These include CDK9 at lysine 48, which promotes this kinase activity and is important for recovery from medication arrest. This site is actually evolutionarily conserved amongst most of the CDKs, so pretty interesting. sirtuin also deacylates ATR interacting protein at lysine 32 that promotes binding to RPA single-strand DNA and activates ATR checkpoint kinase signaling cascade that in turn phosphorylates numerous downstream substrates. We identified a large number of other proteins, BARD1, BRCA1, and HR, homological combination repair, DNA PKCS, and COO, and non-less end joining. So our findings suggest that sirtuin regulates a whole network of proteins involved in DNA damage responses, and dysregulation of this pathway leads to genomic instability and development of cancer. So I just want to highlight one of these proteins that we showed deacylation. This is work by Elizabeth Minton, a talented graduate student in our lab. So she found that sirtuin deacylates the ring domain of BARD1. So this BRCA1 BARD1 heterodimer is at its ring domain, and this is important for tumor suppression, its stability, and interaction and localization. So we identified 3 acylation sites, lysine 96, forms this crystal structure of the ring domain, BARD1 BRCA1, forms a salt bridge with aspartic acid 40 and BRCA1, also lysine 46 and lysine 110, forms electrostatic interactions with glutamic acid and BRCA1. So you can imagine if you have acylation at these sites, there's electrostatic repulsion, and then you have thus loss of this heterodimerization. So we have data here showing this is co-IP analyses. Here we're knocking down sirtuin-1, co-IP of GFP BARD1. We have an impairment interaction with BRCA1. You can rescue this with expression of BARD1 3KR. This is a lysine to arginine mutation that cannot be acetylated. So it shows unequivocally that SIRT1 deacylates BARD1 to promote the heterodimization of BRCA1 BARD1. We have some data showing if you knock down SIRT2, decrease stability of BRCA1, you can rescue with a wild type and 3KR, but not the 3KQ mutant. This is a acetylation mimic mutation. Showing again unequivocally you're rescuing the SIRT1 depletion with BARD1 that it's mediated by SIRT2, the stability of BARD1 BRCA1. And we have some other data showing that SIRT2 can mobilize BARD1 to DNA damage sites. So these findings lead to a model in which SIRT2 deacylates BARD1 at its ring domain that facilitates heterodimerization, interaction, stability, localization double strand breaks, and homologous recombination repair. So we've shown that you can knock down SIRT2, cause sensitivity to a number of DNA damage agents. Can you exploit this for cancer therapy? We use AGK2. This is an inhibitor of SIRT2, NAD plus analog, and we can show hypersensitivity radiation. And then these are A549 non-small cell lung cancer radiation-resistant tumors in mice, and we can delay tumor growth with this SIRT2 inhibitor. So we've shown that SIRT2 is a well-known breast tumor suppressor protein. What is the significant cancer-associated SIRT2 mutation? So Head analyzed the Cancer Genome Atlas for a number of these mutations and used a number of prediction tools, Grantham, SIP, Polyphen, Conserve, determine functional significance. Many of these are evolutionarily conserved, indicating functional significance. So we looked at the structure, and many of these are located in the catalytic site or NAD plus binding pocket. She looked at genomic instability as a readout, spontaneous gamma-H2X or micronuclei. These are SIRT2 knockout cells. You have increased gamma-H2X and micronuclei. You can rescue this with expression of SIRT2 wild type, but not the H187Y inactive mutant. And also these mutants have impairment in rescue, suggesting that these cancer-associated mutations impair SIRT2 activity in genome maintenance, and that SIRT2's role in genome maintenance, tumor suppression may, at least in part, be to its role in genome maintenance. So I want to move on. This is No-Kong Long. He's a new postdoc in laboratory, and when he joined my lab, he wanted to understand what is the role of SIRT2 demeracylation. So SIRT2 it actually has a stronger demeracylation activity compared to deacylation activity. This is very interesting. Not very much is known about meracylation. This is a 14-carbon myristic acid. It's been shown to mediate protein membrane interactions, protein-protein interactions, even cellular signaling. So we used a proteomic approach. This is a myristoyl alkyne probe incorporated in cells. You can use biotin azide, and through click chemistry by copper catalyzed, you form a 1,2,3 triazole using strep-avidin agarose that binds to high affinity biotin. You pull down assay, you trypsinize, do tandem mass spec labeling, and then perform mass spec. So this is in collaboration with Rongku Wu at Georgia Tech. And the Western just shows kind of the validation of this approach. Using myristoyl probe, we can pull down myristoylated proteins, and this is AR1, which is a positive control, a protein that's known to be myristoylated. We can inhibit this with N-myristoyl transferase inhibitor. So we identified over 576 myristoylated proteins. This is more than any other mass spec has been done in the past. In fact, a number of our hits are in common with previous mass spec analyses. We also show that if you use an NMT inhibitor, we can decrease myristoylation at least twofold for 35 of these proteins. We had also overexpressed SIRT2, and you can show 66 of these proteins showed greater than twofold difference in demyrisylation. So we did reactive pathway analyses, found a number of different pathways involved, and a particular interest are those involved in DNA damage response, which is kind of surprising to us. We weren't really expecting many of these nuclear proteins to be myristoylated. We found at least 14 DNA damage response proteins, at least annotated as DNA response proteins. 6 of these are regulated by SIRT2. Some of these proteins include BMN, RPA, Ku70, RUB1. So pretty interesting, very exciting to us. This is still very preliminary work, but we've validated 2 of these hits. This is the myristoyl probe pull down, which we can pull down Ku70 as well as RPA. And we also did kind of reciprocal interaction with, I don't know why this is not showing up here, but Also, we do have validation of the mercenated proteins. Also, the reciprocal direction with IP of 270 showing it's mercelated. So just very preliminary data, but very excited about this kind of new direction for laboratory. So I want to move on to the next section. So DNA double-strand breaks, these are highly cytotoxic lesions, which must be repaired in order to preserve genome integrity. These are paired predominantly by two major pathways. You have non-homologous end joining. This involves kind of direct ligation of broken DNA ends. It's an error-prone process and active in all phases of the cell cycle. And then we have homologous recombination repair. This involves a template in a sister chromatid where you have homologous recombination repair. It's active only in S and G2 phases. It's an error-free process. So that process of homologous recombination repair requires a process called DNA end resection. You need to generate 3' single-strand DNA tails by CTIP or MRN that is then bound by RAD51. And then with the assistance of mediated proteins such as BRCA1 and 2, then it mediates homologous recombination repair. So we've been studying this protein called SAMHD1, serial alpha motif and HD containing protein 1. It's the only known DNP triphosphohydrolase in cells. So it cleaves off this triphosphate group and thus depletes DNPs. It's most well known as an HIV-1 restriction factor. So it depletes DNPs required for reverse transcription. It's also dysregulated in Aicardi-Gouttiere syndrome. This is a congenital autoimmune neurodegenerative disorder where dysregulation of number of DNA processing enzymes such as ADAR1, TREX1, RNase H2. SAMHD1 is associated with CLL, colorectal cancer, and a number of other cancers. And these AGS cells also have genomic instability. So a number of years ago, we showed that SAMHD1 has this non-canonical DNTPase independent role in promoting DNA end resection to facilitate double-strand break repair by homologous recombination repair. It does this by recruiting CTIP to these double-strand break ends, then that facilitates chewing off the DNA ends, and then facilitates homologous recombination repair. So I wanted to highlight the postdoc on the study, Wako Dadacha. He was recruited as a R1 diversity supplement candidate, mentored to a NCI F32 grad, and then subsequent NCI K1 grad, and now has his own independent laboratory at Med College, Georgia. And we have 3 other trainees, Yasmin Castellan, also Famata Sese, and Mark Essian, also on R1 diversity supplements. So we've shown that SIR2 has, SAMHD1 has this DNA end resection role. It also has this kind of DNA role, and they're kind of disparate functions. So what might regulate SAMHD1? Priya Kapoor in my lab investigated the acetylation of SAMHD1 and showed that it is indeed acetylated. You can IP SAMHD1, do Western, you can see acetylation. Add radiation or camptothecin, you can see a decrease in acetylation. This is specific. You don't see any deacetylation in response to UV or MMS. They're very specific to double-strand breaks. We can do rescue assays, knocking down the nuclear sirtuins, knock down SIRT1. You can see alleviation of this deacetylation, but not with 2, 6, or 7. And then you can go do co-IP analyses showing that the SAMHD1 complexes with SIRT1, but not 2, 6, or 7. So these data indicate that SIRT1 interacts with and deacylates SAMHD1 in response to double-strand breaks. So what sites might this be deacylating? We performed proteomic analyses, identified a number of putative acetylation sites, generated lysine to arginine mutations that cannot be acylated. So you see, if you mutate lysine 354 to arginine, most of the acetylation is decreased. We also see some decrease in acetylation at lysine 4/5, but we see a continued deacetylation in response to damage, indicating that 354 is a major site of deacetylation in response to double-strand breaks. We can validate this by generating an acetylation site-specific antibody toward lysine 354. We can show that endogenous SAMHD1 is deacylated in response to radiation treatment. We can rescue this by SIRT1 depletion. So what is the functional significance of this deacetylation? So we performed this well-characterized direct repeat GFP or green fluorescent protein homologous recombination reporter assay. So we have cells that have an integrated reporter substrate. GFP is mutated. You express this rare kinase endonuclease, which induces double-strand break. And only if there's homologous recombination impaired, you have expression of GFP you can detect by flow cytometry. So using this assay, you knock down SIRT1, you see a decrease in homologous recombination repair. You can rescue with wild type and 354R, but not 354Q, which is the acetylation mimic. So this shows unequivocally that SIRT1 deacylates SAMHD1 at 354 to promote homologous recombination repair. We don't see full rescue, likely because SIRT1, we're rescuing SIRT1, not SAMHD1, likely because SIRT1 deacylates multiple other substrates involved in HR. We also looked at end resection, phosphorylation RPA32 at serine 4, serine 8, knockdown SIRT1, you see an impairment in phosphorylation, rescue with a 354R, but not 354Q. So here you see more of a rescue than the HR. SAMH1 is a tetramer, has DNP activity. So we also kind of analyzed these functions in collaboration with Eric Ortland using size exclusion chromatography. Really no difference with the wild type versus the mutant. We also looked at DNP pools for DNP's activity, collaboration with Baek-Kim. This is a DNP primer extension assay developed by Baek-Kim. You overexpress SAMH1 or these mutants and really no impairment. So we did look at localization and knockdown SIRT1 or inhibit SIRT1. You see impairment in SAMH1 localization to these radiation-induced double-strand breaks. We also have this Falk1 mTERA system developed by Roger Greenberg, where there's an array of double-strand lac operons, and you can induce Falk1 expression, and that induces about 250 double-strand breaks at a very localized site. And we see that 354 localizes there, but not the 354Q, indicating that 354 de-escalation by SIRT1 facilitates its recruitment to double-strand breaks. So SAMH1 is known to bind to DNA. We did these single-strand and double-strand DNA with 5' overhang pull-downs, and we can see that the wild type and KR bind better than the KQ mutant. So this is with cell lysate and determine whether this is a direct binding. We generated a peptide, 332 to 384. We acetylated on one of them, and this is electrophoretic mobility shift assays in collaboration with Shiming Deng's lab, and showed that only the non-acylated form, you see a shift in mobility because due to binding to the free oligos, indicating that there's a direct binding that 354 mediates between SAMH1 and single-strand DNA. So SAMH1 recruits CTEP to DNA damage sites. We can also show that 354 is important for mobilizing CTEP to binding to single-strand DNA sites. You can see this impairment in binding, SAMH1 knockdown, rescue with wild type and KR mutant, but not the KQ mutant. So our findings lead to a model which responds to DNA damage, SIRT1 de-escalates SAMH1 at lectin 354 that facilitates its binding to single-strand DNA, which in turn recruits CTEP, promotes the end resection, and facilitates small inflammation. Okay, so can we target SAMH1 for cancer therapy? So HIV-2 has this viral accessory protein called VPX that targets SAMH1 for proteasomal degradation. So we wanted to kind of take advantage of this natural way of degrading SAMH1. We packaged VPX in virus-like particles. It basically contains the structural proteins can't replicate and kind of a delivery tool to deliver SAMH1 cells as well as tumors. And we can show that with these VLP VPX, we can degrade SAMH1, cause hypersensitivity to camptothecin as well as PARP inhibitor. This is a specific effect because we can rescue by overexpression of SAMH1 GFP. So using a very natural way of exploiting how HIV-2 degrades SAMH1 for cancer therapy. Can we target SAMH1 in breast cancer? We analyzed a number of breast cancer cell lines. The triple negative breast cancer subset has very high expression compared to non-tumorigenic cells, suggesting that there's a potential for a therapeutic gain. We also analyzed the Cancer Genome Atlas, showing that low SAMH1 is associated with better outcomes. So this is in triple negative breast cancer cells at SAMH1, VLP VPX, degrade SAMH1, cause sensitivity to PARP inhibitor. And we also have some data we can do direct intratumoral injection to degrade SAMH1 in vivo and tumors. So this is a collaboration with Zach Buchwald, very talented junior investigator at our institute. He's a cancer immunologist. So this is a recent funded DoD breast cancer research program grant. So he's found this immunological niche consisting of antigen-presenting cells in close proximity to stem-like CD8 plus T cells, and shown that when you have higher niche proportion, this is associated with better outcomes. SAMH1, in addition to acting at double-strand breaks, also acts at stalled replication forks and degrades this single-strand DNA, which otherwise goes to cytoplasm, and through the C-gasking pathway, induces innate immune response that potentiates immune checkpoint inhibition. So we're investigating how SAMH1 might regulate progenitor stem cell differentiation, as well as using VLP VPX strategy for potentiating immune checkpoint inhibition in breast cancer. All right, so I wanted to kind of move on to our next project. Most of this is unpublished data. CH5 is chromodomain helicase binding protein 5. It's got two pH domains that bind histones, two chromodomains, a SNP2 like ATPase helicase domain, domain of unfunctional significance, and a CHD C-terminal domain. Ali Maysmill's lab has shown that it's a dosage-dependent heterozygous tumor suppressor can form neuroblastoma, squamous cell carcinomas. It's a nucleosomal remodeling complex component. So this project begun quite a while ago, in 2014, by very talented resident Bill Hall. He's now like a full professor at Medical College of Wisconsin. So what Bill did is he analyzed cells that had depletion of CHE5 and showed that these increased markers for phospho-CHEK2 as well as damage drugs, markers for DNA damage response. And then he analyzed these pancreatic cancer cell lines and showed that those with low CHE5 expression had increased markers for phospho-CHEK2 and damage drugs. So low CHE5 increased activation DM damage response. And then collaboration with Shisher and myself, we analyzed pancreatic cancer patients treated with surgery followed by adjuvant gemcitabine-based chemotherapy. We did IHC for CHE5 expression and showed that those with a low CHE5 expression were associated with worse outcome. So we interpret this as those with low CH5 expression have increased genomic stability, and thus these cancers are more aggressive and thus do worse. So I had a number of lab techs kind of work on this over the years and kind of not really get very far. Finally able to convince a talented postdoc, Sandeep Roth, to work on this project. This is also a collaboration with Lee Lan. And Sandeep showed that you knock down CHE5, we have hypersensitivity to etoposide and bleomycin, which induce double-strand breaks, but not UV or MMS. These knockout cells have hypersensitivity to radiation, can rescue with the wild type, but not the DEAA calcium active mutant, suggesting that the helix activity governs resistance to double-strand break. So we also look at localization. This is the FOC1 mTERA system again. CH5 localizes these sites. We also use Gaio Lagoub's DIVA system cells expressing a CC enzyme, which induces about 150 double-strand breaks at annotated sites in cells. We also, so it has very nice localization to these sites, co-localization. We also did a chip quantum PCR showing a direct binding of CHE5 to these double-strand break sites. So what localizes CHE5 to these sites? We inhibited several upstream regulators, ATM, ATR, DNAPK, basically no impairment localization, but then you use PARP inhibitor, you see this really nice impairment localization. We also see knockdown, when you knock down PARP1, PARP2, impairment localization CHE5 to these sites. We also have some QIP data, pretty interesting CHE5 complexes with PARP2, but not PARP1. Really not sure why, but it's pretty interesting. It's not regulated by DNA damage. You can see no difference interaction with radiation, not selectively regulated, unsynchronized with S2, same interaction, as well as it's preserved after benzonase nuclease treatment, so it's not mediated by DNA. We can also knock down PARP1, PARP2, and show impairment in binding of CHE5 to double-strand breaks. So PARP mediates chromatin relaxation as well as PARylation of proteins to kind of distinguish between whether CHE5 might bind to PAR chains or mobilize the sites due to chromatin relaxation. Instead of adding PARP inhibitor before the induction of damage, these are laser microradiation sites, we added after the induction damage. So with a control RNF146E that's known to bind to PAR chains, you see kind of this resolution of this localization to these damage sites. In contrast, CHE5, you see no resolution, indicating that the PARP-mediated chromatin relaxation facilitates its localization to these damage sites. We can validate this by overexpressing ALC1 wild type, which is a chromatin remodeling protein, but not its calcium inactive mutant. There's a collaboration with Sebastian Wett. So CHE5 is important for radiation resistance. How might it mediate this? This is a well-characterized DRGFT reporter assay again, and you knock down a CHE5 and you see impairment HR, rescue with wild type, but not the DEAA mutant. We also have a similar a construct for non-homogeneous end joining, an EJ5 assay, and here showing knockdown CH5, we actually see an increase in non-homogeneous end joining. We can validate this with iron-induced rapid lymphocyte, which is impaired as a marker of HR, and RIF1 is a marker of NHEJ, which is increased. This is pretty interesting. It suggests that CH5 plays this role in double-strand repair pathway choice between promoting homozygous repair, which is more error-free, versus NHEJ, which is more error-prone. So HR requires kind of this DNA end resection process. So we looked at BRD exposure kind of under non-denaturing conditions. And only if you have resection do you have exposure to the BRD that can be recognized by antibody. So knock down CG5, see impairment in end resection, rescue with the wild type, but not the DA mutant. And we can also validate this by a PCR-based DNA end resection assay. We also looked at kind of downstream regulators, CTIP, knockdown CG5, see impairment in CTIP localization to these damaged sites, as well as impairment in CTIP binding to these double-strand breaks by chip quantitative PCR. So CG5 is a chromatin remodeling protein. We want to determine whether it might play a role in remodeling chromatin to facilitate recruitment of proteins. So we did laser microradiation of PHFP2H2A. You can see over time, you see this kind of expansion of this microradiation site. And with CHD5 knockout cells, we see impairment in this expansion. And we also validated this with a micrococcal nuclease-based assay, PCR-based assay at annotated double-strand repair sites. So we performed kind of proteomic analyses looking for various histone modifications. We're still analyzing this, and I'm just reporting 3 of these sites that are very notable. Histone H3K36 that recruits CTEP. To knock down CHD5, we see kind of an impairment in H3K36 prime methylation. And rescue with the wild type but not the DA mutant. On the other hand, if you look at histone H4K20 di and trimethylation that recruits 50BP1, which is involved in non-mod end joining, we see an increase in this modification. So how might CH5 be doing this? We looked at the writers for these sites, SETD2, which methylates H3K36 trimethylation, and you knock down CH5, you kind of see that impairment in SETD2 recruitment to damaged sites. This is not due to expression of SETD2. We see kind of no difference in kind of expression levels. On the other hand, you look at KMT5B, knockdown CH5, we see an increase in localization of KMT5B, or rather binding to double-strand break sites, and no impairment in total expression of KMT5B. So we've shown that CH5 plays kind of this role in double-strand repair pathway choice. This is lead general stability. In knockout cells, we can show an increase in 50p nuclear bodies, which is a marker for genomic instability. And we also can see using chromosome breaks, many more in these knockout cells. So our findings lead to a model in which, in response to double-strand breaks, part mediates chromatin relaxation that recruits CHG5, that in turn mediates chromatin relaxation that recruits SETD2 to methylate histone H3K30's trimethylation that recruits CTIP and promotes end resection and homologous repair. In CHG5 deficiency, you have KMT5B that's recruited that methylates histone H4K20 dimethylation that recruits 50pp1 for non-homologous end joining. Okay, so I just want to finish off because we have this NCI U54 Robin Award. This is Radiation Oncology Biology Integration Network. So the foundation of this concept is that there's very little data for radiation resistance in human patient samples. Most of it's in cells as well as tumors. And so the NCI wanted to fund several centers to get some more data on this. And the principle is to get data before, during, and after radiation treatment, very dense collection of tissue. And we do various kind of omic analyses, single-cell RNA-seq, deep exon sequencing, immunologic profiling, spatial transcriptomics, and circulating DNA, as well as kind of very dense imaging analyses. And we're generating longitudinal molecular characterization data sets for further analyses. There's also a workforce development cross-training core component of this, where we're promoting interdisciplinary team science, as well as an engine for kind of spin-offs. So the structure of this U54 is radiation oncology, clinical, medical physics, radiation biology, radiobio, and kind of synergistic interdisciplinary team science. 3 of these centers were awarded. We all kind of teamed up. Tim Chen and I, Clinical Clinic, are multi-PI on one of these centers. So this is an overview of our U54 GenRad Center, genomic, microenvironment deterrence, temporal dynamics, and efficacy of radiation-based combination therapies. Tim and I are multi-PI on this award. Xiaofeng Yang and Omar Mian run the cross-training core. There's 2 molecular characteristic trials, one bladder cancer, one driven by Omar Mian and Shobha Gupta, This is basically radiation with SASE-govitecan and muscle-invasive bladder cancer ineligible for concurrent cisplatin-based chemotherapy. There's a head and neck re-irradiation study driven by Nabil Saba, re-irradiation with concurrent nivolumab in patients with local regional recurrent or secondary primary squamous cell carcinoma of the head and neck. This also involves Nikki Schmid, Zach, and Bill Stokes. It's a transitional and clinical core driven by Hayden Kisek. And there's three major projects. Zach and I run third project or drive the third project, looking at molecular mechanisms underlying therapy response to radiation and immune checkpoint blockade. My sister Jen is also involved in project 2. So these are the two major trials. Adaptive radiation therapy with concurrent SASE-govitecan for bladder preservation in patients with muscle-invasive bladder cancer. The eligibility criteria: muscle-invasive bladder cancer T2, T4A. You're doing maximal QRPT and then concurrent adaptive radiation and SASE-govitecan. We're getting circulating tumor DNA, urine cytology, biopsy before, during, and after radiation treatment, and doing very kind of dense collection of tissue. This is the head and neck re-irradiation study. The eligibility: recurrent or secondary head and neck squamous cell carcinoma, overlap with the prior RT field of greater than 40 gray, RPA class 1 and 2. We're doing nivolumab with concurrent radiation therapy. We also have a standard of care arm with radiation and concurrent cisplatin-based chemotherapy. And we're collecting tissue before, during, after treatment, circulating tumor DNA, oral cytology, and also very dense imaging analyses before, during, and after treatment. This is Nabil's study presented in abstract form, showing that re-radiation with nivolumab has very good control rates compared to historic controls. It's about 2 years, about 30% overall survival rate. Our project 3 is molecular mechanisms underlying radiation plus immune checkpoint blockade versus radiation plus cisplatin, co-led by Zach and myself, also involving Rafi, Nabil, Chalfon, Tim, Nikki, and Bill Stokes. The rationale for studying head and neck cancers is that 75% head and neck squamous cell carcinomas are treated with radiation therapy, but many of these recur, and the success rate of re-radiation is quite poor. Immune checkpoint blockade is a potential therapy option, but only 15-20% response. So there's a need to understand mechanisms for resistance to immune checkpoint blockade as well as radiation in the recurrent setting. This is data from Rafi's lab, single RNA-seq, deep profiling of antigen-specific CD8 plus T cells in head and neck squamous cell carcinomas, HPV positive, identified a number of clusters, stem-like, transitory, and terminally differentiated. Since Zach's data showing that higher tumor niche proportion is associated with better outcomes. And Zach's got some data showing that radiation and anti-PD-L1 can synergize to promote progenitor stem-like CD8 plus T cell tumor infiltration. And you can look at these tumors, you can see delayed tumor growth with this combination, both at the primary site as well as the abscopal site. And there's this whole body of data showing this interplay of DNA response with tumor microenvironment. Here showing that when you have dysregulation of BRCA1, BRCA2, you see differences in progenitor stem-like CD plus T cell populations. So we're very interested in understanding what these mechanisms are, the interplay of DNA response and response to radiation, as well as cancer immunology. And finally, I want to just acknowledge members of my laboratory, because we're only good as our trainees. This is my lab kind of over the years. I like to have fun. We do these Halloween parties kind of every year. I also want to note, we've had a number of trainees come through the lab that have since entered clinical oncology. Daniel Tenenbaum, Man Rudra, Vishal Deer and his wife, Erin Connolly, actually Slotstein, Jake Wynn is somewhere, Neil Pfister, and also some other trainees that have ties to the department, Dinette Danelia, Jimin Jung, and Nikita Dang. We have a number of other collaborators that we're very grateful for the contributions, and happy to take questions.
Thank you, Dr. Yu. If you have any questions, please use the QA feature located at the bottom of your screen. While we wait on questions, I wanted to mention that next week we have an exciting talk from Dr. Diwakar Davar from UPMC presenting on the inside scoop on poop, intestinal microbiome in cancer immunology. Dr. Davar has assured us that he will come with, quote, an infinite number of poop jokes, so please be sure to attend next week's talk. To view all upcoming Winship Grand Round lectures, please visit the Grand Rounds page on Winship Cancer Center website or the Winship calendar. And any questions while we
Yes, Alex.
I have a about 91 maybe.
Yeah.
It's supposed to be a very good market for psychology and responses and there.
Correct, yeah.
I guess my question that I'm not too sure that by providing an 81 that favor the response to one type of therapy to solution. In other words, it's mental to others. Like that, I also type of therapy and
Yeah. Yeah, very, very good point. So SAMH1 is a DNPase, so also metabolized kind of cytarabine, which is kind of an analog. So you can imagine we're silencing SAMH1, you have sensitive radiation, but then you're also maybe having worse outcomes to other chemotherapeutic agents. So yeah, I think we would need to determine what specific types of cancers SAMH1 might be more responsive to. I don't know. I think probably high expression is certainly like one of them. If there's greater dependency on SAMH1 DNA and resection role versus its role in DNPase role. So I think our marker, for instance, and our lysine 354, we have an acylation site-specific antibody that could potentially be used as a biomarker, kind of distinguish response between those two different treatment modalities. Yeah, Ron.
David, a big level, right, although science. You talked about first two being a tumor suppressor.
Right.
And then you also showed a strategy where inhibiting it actually would be helpful in treating cancer. So do these two conflict against each other? How do you understand that?
Yeah, no, I don't necessarily think so. I think there are a lot of like tumor suppressor proteins like BRC1 or BRC2 where we're still targeting double-strand repair pathways to potentiate. So you can imagine maybe like over time you have search deficiency, creates genomic instability, and then greater dependency on certain mechanisms of repair that then you can then target. So I don't think it necessarily you can't reconciliate those two kind of disparate strategies. Yeah. That's correct. Yeah, so whether it's to its catalytic activity or kind of a non-catalytic role has not been investigated, and that would be interesting because there are differences. Shanja's got some interesting data showing you have kind of non-catalytic role for various PIKK proteins. Larry.
Maybe I'm going to follow up on that. So if you can combine markers inhibitors, either loss of HER2 or HER2 inhibition in a similar way to have the problem.
Yeah, absolutely. I mean, in fact, we have that data. If you knock down HER2, you have HR deficiency that sensitizes the PARP inhibitor. Oh, is there a chat box? Okay. Ned Waller says, impressive tour de force, the biochemical machine responsible for DNA repair, looking at the dose response of radiation and quantitative cell survival looks like the search and knockout lacked the shoulder to repair and cell survival associated with low dose radiation. We showed that PI3K inhibitors can upregulate search in T cells, suggesting that chronic activation of the AKT mTOR pathway can cause senescence and aging of T cells, and a synergy of agonists in the PI3 AKT mTOR pathway and radiation. Yeah, I certainly I think so. I'm not familiar with all of that data, but certainly I think certainly would be very possible. If there's anything else, I think that's probably it.
you again, after you, and looking forward to seeing everyone next week.